It's weird, huh? Yeah, it too. Okay, so today we're going to finish out at the end of our membrane transport part 3 packet. So we're going to finish talking about osmosis. We're going to talk about a couple ways we can think about tying together these different types of transport as we're moving across different layers of tissue in body. And then we'll talk a little bit about exam format and studying. On Monday, we'll have some time to review, maybe get a quick peek at some things that are coming up in our next unit, depending how into review people are. But definitely content on the exam we'll only go through today. So, and quick show of hands, is anybody able to see the practice exam I put up? There should be one up at, okay, cool. I hadn't seen if anybody clicked into it, so I wanted to make sure it was visible. Awesome, so we'll talk about that too. Okay, so when we left off, we had introduced our concept of osmolarity, so we can measure solute concentrations in a way that helps us focus on thinking about water and how we affect water movement, which is really important when we're thinking about cells, when we're thinking about human tissues. So we had looked at how we convert that. We take something like glucose that doesn't split up in water. We're going to have similar numbers for our molarity of a solution and our osmolarity of a solution. But when we have salts or things that are ionically bound, they separate. We're going to have to double that when we think about the effect on water. We linked up those measurements to the idea of osmotic pressure, so that these concentration differences, these solute differences are actually creating a pull on water that we could conceptually figure out how much force it would take to stop, which is what our osmotic pressure is. Okay, so we had left off here looking at the difference between osmolarity and tonicity. So osmolarity being for measuring those concentrations that affect on water, but tonicity focusing on the fact that some things can move across cell membranes, but other things can't. So focusing on the impermeant solute. So we had seen this example where we start out in image A with an example that is isoosmotic. It says that up top, we talked about it, right? So we have the same sort of concentration of things inside and outside the cell at the beginning. This is a situation in which we are actually hypotonic, right? Right, so this highlights the difference between those two things. And the reason there's a difference is because in this case, our yellow solute can move through cell membranes, so it's permeant, but our purple solute is impermeant, can't move, membrane isn't permeable to it, so it's the thing that's stuck. So ultimately, because our permeant solute is going to move down its own gradient, we're going to end up with a situation where water specifically follows that tonicity, right? So ends up moving towards the center of the cell, where we're going to end up with a higher osmolarity at the end because of that movement of our permeant solute, and then water flowing towards that higher osmolarity. So these things are linked, but slightly different. We can just sort of narrow our view and look at the tonicity specifically, just our impermeant solutes if we're just trying to predict water movement. Right? We don't necessarily need to look at those little yellow particles if we just want to know where water flows. Water is going to flow ultimately towards where the impermeant solutes are. But this is kind of a two-step process as to why. So here we have some terms measured out, or described out, relating those osmolarity and tonicity concepts. So we have similar terminology for both. So we have those prefixes, iso, hypo, and hyper. Iso meaning same, hypo meaning less, hyper meaning more. So if we're talking about something that's isoosmotic in your body, that's usually going to be something that's 300 milliosmoles. That's going to match what we have in our extracellular fluids, what's in our intracellular fluid, but that would include both permeant and impermeant solutes if we're talking about osmolarity. The difference when we talk about tonicity, we're thinking about impermeant solutes specifically. So we change our terminology a little bit, keep the prefixes, the iso, hypo, and hyper, but now we're talking about tonicity, specifically things that aren't going to cross the membrane. And that's going to be ultimately the more important one when we think about movement of water. It's going to be the thing that gets us to this prediction fastest. So here, looking at tonicity a little bit, when we think about the movement of water, that actually has an effect on cell volume, right? So water takes up space. When it moves, cells are going to swell, cells are going to shrink, depending on the direction of that water movement. And the ratios actually match up pretty well to our measured osmolarity or tonicity, in this case, of our solutes inside and outside the cell, so intracellular fluid versus the extracellular fluid. Okay. So in this case, in the top row, we're looking at a situation where we have 300 milliosmoles inside the cell, 150 milliosmoles measured outside the cell. So in this case, we are in the ECF, the extracellular fluid, or in this case, the contents of this vial. We're in a hypotonic solution. So hypo, less, that number is lower outside. Now, specifically, if we look at the ratio inside and outside, we can see that we have about twice as much of that osmolarity, that tonicity inside the cell, right? So twice as much solute, essentially, in terms of its concentration inside the cell versus outside the cell. So we know that water moves to water down those areas of high solute concentration, get them to space out, trying to put us to equilibrium so that our concentration is the same inside and outside the cell. And when it does that, we can see that the effect on cell volume is actually the same as that ratio if you focus on the inside versus the outside, right? So in this case, we had twice the tonicity, twice the osmolarity inside the cell. After movement settles, water moves into the cell. We reach this sort of equilibrium state where we're now in an isotonic solution, right? So these are now the same. In order to do that, we had to double our cell volume. So water moved until we had twice as much. Yeah, until the cell swelled to twice its size. All right. So we can see that 2-to-1 ratio of the inside to the outside in this first part. And now we have another 2-to-1 ratio, but in this case, it's our new cell size to our old cell size. We can see that happening also when cells shrink. Okay, so in the case where we are in a hypertonic solution, meaning we have more extra excess tonicity outside, right? We're going to see water moving again towards the area where we have a high amount of solute concentration, right? So we are moving to water down that outside of the cell. That's our water movement. And so being pulled towards this higher tonicity, this higher osmolarity. And we can see that in our original cell, we had are inside of the cell, measured at about half the concentration outside the cell. Right, so we have 300 versus 600. And we can see that we have that same relationship when we look at the cell volumes now. So our cell from this first image to the second image to get us to this isotonic situation, halved in size. Okay. We keep the same sort of ratio. We can predict how big or how small cells get as we see water moving across these gradients. Okay. That's what we want to know about osmosis right now. So we have aquaporin proteins in our cell membranes. They allow water to move. We're assuming they're in every cell unless I tell you otherwise. They move in relationship to solutes, in particular impermeant solutes, so things that can't move across the cell membrane. They move towards regions where we have higher solute concentrations, measured by this higher osmolarity, enacting a pull on water, which can be measured as osmotic pressure. So now we're going to link some things together to talk about not just how do things move into or out of one cell, but how can things move across like a layer of the body. So we're thinking here, transport across epithelium. So epithelium being these linings, so like the skin, although in this example, we're going to be walking through a lining in the gut tube. But we know that our different body compartments are separated out by these epithelial membranes that kind of seals. Okay. So we're going to have to transport across a cell. When we say across a cell, I don't just mean into the cell, right? We're going to be thinking about how do we get movement from one side of the cell across one membrane, and then also again across a second membrane, right? So we now understand how we look at the forces driving something into or out of cells, but how do we link those two things together? Right. So we want to do a little bit of labeling a little bit of vocabulary as relates to cells right here to make sure that we can all describe this. Hopefully you've seen some of these words before, but if not, I'm just going to point them out. Okay. So first going to point out some regions that aren't on the bullet points, right? So what we're looking at here on top is the lumen of our gut tube in this example. A lumen is just a hollow space, so you'll see that sometimes. Here we see our interstitial fluid on the bottom. So we've mentioned before the ISF. That was our interstitial fluid, one of our two types of extracellular fluid, specifically the type of fluid that we see surrounding body tissues. And our other one was the plasma, which we saw specifically in the blood. Okay. So usually when we see extracellular fluid around cells, this is the specific type of extracellular fluid we're talking about. Okay. So looking at the cell itself that's in this environment, we can see that neighboring cells are linked together by these tight junctions. Right? So these are our tight junctions, these little clam shells here in between. What a tight junction does is a feature that we see in epithelial membranes, epithelial tissues. They form a tight seal, so they're not going to allow things to pass through and avoid going through a cell, right? We're not going to see things going through tight junctions. Purpose of a tight junction is to make sure that we have to regulate whatever crosses this epithelium, right? So we're going to have to force something through one membrane and then through a second membrane if we want to get to the other side of the epithelium. Okay, so that's the tight junction piece. Then we have some direction words. So when we're talking about like top and bottom of this image, we're going to describe this a little more formally. So the apical membrane, the apex of the cell, in this case, the top of the image is the one that's facing outside. So we're remembering that the lumen of the gut tube, so like the hollow part inside, counts as part of your external environment. Not from a colloquial English perspective, it's inside your body, but from a physiology perspective, it's not, right? We haven't processed it, we haven't regulated it, it's still continuous with the external environment. Right, so our apical membrane is going outside to this hollow space, this lumen, and our basolateral membrane, baso for the bottom, lateral for we're sneaking up the sides here, we're sneaking up laterally as well. And this is always going to be on the blood side, right? So this is the internal environment, this is in towards your body, basolateral, that B matches the B in blood, right? So basolateral membrane is going to be on the blood side. At the bottom of the side. Okay. So we're going to be thinking about how we transport solutes, like from nice salty French fries that we ate that are in the lumen of our gut tube into our bloodstream, right? So we're thinking about solute transport. Here we can see some sodium up here in the lumen. We can see that ultimately the sodium ends up transported into your body and ultimately into the bloodstream. We're going to think about how that happens. We're also going to think about what else can we do with this sodium gradient that we're creating. So what we're going to notice on this image is that we have a couple of different types of membrane proteins. So focusing on this example, what we see on the basolateral membrane, in this part of the image, we can see that we have a sodium-potassium pump. Right? So remember, a pump uses ATP for energy. This is because it's moving something against its gradient, against, in this case, because we're thinking about ions, they're electrochemical driving forces. So we need a source of energy to push. Okay? And this pump is doing primary active transport because it's directly using the ATP. Right, so we can see in this case, for example, we're moving sodium from an area where we don't have many sodium to an area where we have a lot of sodium. Right, so what we're observing is that sodium in this case is going from low concentration to high concentration. Conceptually, we can link that up to our slightly more complicated understanding of electrochemical driving forces, which would work the same way. So we know that they should go from high to low in our favorable direction. So to pack more sodium outside the cell, we have to put in energy, is a way we could notice this. So that's our primary active transport piece. We can see we also have a potassium channel here. Okay. So in this case, in our potassium channel, I mentioned that your book likes to use green for passive transport, for favorable direction. So we can see that we have the potassium moving through a channel protein, a channel that's open, as we can see in this image, in the favorable direction. So this is passive transport through a channel. Okay? So here we can see that the potassium, when we're looking at what's happening with respect to the channel, is going from an area of high concentration to an area of low concentration, like it should, right? That's what it wants to do. This channel is open, like it is in the image. That's the direction we see movement. That's the spontaneous direction. Okay. Now, one of the things that we're going to be interested in when we think about that, when we think about eating, when we think about absorbing, how do we get energy from our food, is glucose. And we've talked about metabolism, we've talked about how we break down glucose for energy, ATP. So as we walk through this, we're also going to see a glucose carrier on this basolateral membrane. It's not in this image right here, but we're also going to have something moving glucose across that way. I'll do that in a second. There we go. Yeah. So it's in this larger image. just wanted to walk you through some bits of this. So some of this is the same. So we had the sodium potassium pump we already looked at. We already looked at this potassium channel. So this would be an example maybe of a leak type channel. We see potassium being actively transported into the cell by the sodium potassium pump, but then we see it leaking back out passively through that channel. Okay. Now we're adding on the glucose stuff. Okay. So here we see that glucose carrier on the basolateral membrane that's mentioned on the previous slide. Okay. So we got another green arrow. This is another type of passive transport. So this is that facilitated transport. So we have a carrier protein, right? So we can see this little purple circle, right? The glucose isn't going through it, but the glucose is being carried by it. Passive process because it's in a favorable direction. We're going from high glucose concentration to low glucose concentration. Right? Not as dramatic as with the sodium, but high-low gradient here. So this is a favorable, passive, spontaneous direction, doesn't require energy. The carrier just happens to change shape due to coming into contact with glucose, flipping over, carrying it across. Okay. And we can also, in this image, see what's happening on the apical membrane. Part of why it's so busy, we've also added stuff up there. Okay. So on this apical membrane, we can see that we now have another pump. Right? Right, so here's another pump. So here we see that we use a sodium gradient. Right? So we use a sodium gradient going in its favorable direction. And so if we're looking at sodium, got high sodium here in the lumen of our intestine. We've got low sodium inside the cell. And so that's a direction sodium likes to go, high to low. Great, moving our correct favorable direction. But we're pairing that up with the movement of glucose. So if we look at the other side and we're looking at that glucose concentration now across the apical membrane, our glucose in the lumen is relatively low. We're packing all that glucose into the cell. Okay, so here our glucose concentration is high. Oh no, we're going low to high. That's why this needs to be a pump. That's why we need to use that sodium gradient to carry glucose along with it across our apical membrane. This is an example of secondary active transport. Right? So secondary active transport is when we use the concentration gradient for the movement of one molecule in a favorable direction to move another molecule in a less favorable or unfavorable direction. So we don't directly see ATP, but we did just walk through how ATP was involved in this, right? Our first step would have been depleting the inside of the cell of sodium, right, by pushing it out the other side, right? So we set up this sodium gradient by making the inside of the cell super low in sodium. So if we're thinking about active transport in that 1-2 step way, right? Step one was set up the sodium gradient with this pump on the basolateral membrane, and then step two, secondary active transport, use that pump at the top to bring in glucose, use that sodium gradient. Yeah. So in the real case, there would also be a sodium channel involved here a little bit, but not necessary for linking together these different processes. I think I see it. Okay. All right. So this is one of the complicated ways we would expect to see real transport happening inside the cell. So we're going to see forms of passive transport happening. We're going to see multiple forms of active transport happening, all working together. In this case, our real goal, for the most part, was to get glucose into the blood. Right? when we focus on this example, that's one of our main points of focus is how do you make sure that you actually absorb as much energy from your food as you can, right? How do you make sure you're soaking up all the glucose? And this is how. The sodium is related because we set up this sodium gradient to power our secondary active transport of glucose, right? So we created the sodium gradient using this pumping on the basolateral membrane to power this transport at the top, right? So here we're piecing this out piece by piece in the text on the slide, right? So it's pointing out here in words. Make sure that you notice that we've created that low intracellular sodium using that sodium potassium pump. Right? In the first image that we saw before we added all the pieces, we saw that if we had a sodium channel, that would have drawn some sodium passively into the cell, right, across that apical membrane. Right? So if we were thinking about this, how would you get salt into your body? We could have stopped there. But then we added on this piece of how do we use a salt gradient, a sodium gradient, to move in something like glucose. Okay? So that's adding in this glucose transport here, where we have our secondary active transport using this sodium gradient, and then allowing that to pack glucose into the cell, giving us a high concentration here inside that we can then allow to move passively into the interstitial fluid here and therefore entering the body essentially. If we wanted to add on, we can see that these things, these molecules, the glucose and the sodium ions then enter the bloodstream, in this case, down their gradients as well. So that's how you get stuff from your food into your body. Now, movement of water across multiple membranes similarly would be kind of multi-step. Okay, so this is messier than we usually see because we're usually not drawing in quite so many solutes. We're not focusing so much on water in typical images, but it's important to make sure that we understand what's happening. So in this case, as we're thinking about, okay, if I drink a smoothie or drink a glass of water, how am I getting that water into the body? Right, here we see that the first step of that actually doesn't have to do with the water itself. It has to do with the solute. So here we can see active transport. Right? So active transport, probably primary active transport of a solute. Right? So the primary, the one active transport is that red arrow. Right? So this is creating a high solute concentration in this interstitial fluid, right? So we packed lots of ions into this interstitial fluid. So we now have a gradient, right? So this means that we now have a lot of solute here, right, in these spaces. So here on the side of the basolateral membrane, these sort of projections we see here are sometimes called paracellular spaces, para meaning next to. And so you can see you have high solute concentration here as well. And high solute concentrations have high osmolarity, high tonicity, right? So they draw water towards them, right? So in this case, let's focus on the basolateral membrane first, right? So you can see that we've created a solute concentration where we have lower solutes inside, higher solutes outside, right? So this is essentially creating hypertonic extracellular fluid, right? Hypertonic over here. Okay. Which means that we have across the basolateral membrane that water flowing into that interstitial fluid, right? Now, if water is flowing from the inside of the cell to the outside of the cell, right, this would be concentrating down the solutes that remain or we'd see the cell shrinking. We don't want that to happen. We don't want our tissues to get shrivelly and dehydrated, especially not in like our intestines. So we also have movement of water coming into the cell to replace it from the other side. So if you look at the arrows up top, you can think of this as kind of a 1-2 step, right? So we have water moving out of the cell into these side spaces, these paracellular spaces, and we can see the water flow that's chasing it and replacing it so that our cell stays the same size instead of shrinking. So this is how we would get water into our bodies across a cell, across two membranes. So the overall effect is that using this process of osmosis, understanding how water flows with respect to these impermeant solutes, we get water being transported all the way from the lumen. So that tube that has all the water, the smoothies, whatever that you drank, into our bodies in this regulated kind of manner by regulated state. So an example of kind of deficits that can happen in this process with real life implications come in your book in the example of the disease cystic fibrosis, which is relatively common. I have a cousin with this. So in a normal healthy lung, we have a process similar to what we were just seeing in the gut, but in reverse, right? So in this case, we typically see sodium movement into our airway lumen, right? So into the lungs here. So that's an ion. And we also have chloride movement following that sodium into the airway lumen of lungs, right? So essentially what we do is build up high osmolarity, high tonicity, right? We're in air, but still in this location. Osmolarity for us. Okay, so that exerts a pull on water. So here we see water following that solute gradient to the surface of the lung tissue here. And one of the functions of this process is that it keeps the mucus that lines your lung tissue watery enough that it moves around, doesn't get in your way, kind of like smooths out, stays thin enough across the whole layer of your lungs. Because we want to keep our lungs moist, but we don't want to fill up the space. Okay, so that's what should be happening. But in the case of cystic fibrosis, we got a problem. So what you might observe if you've ever met a person or a patient with cystic fibrosis is that one of the noticeable things they have a problem with is breathing. And part of this is that they have a really thick buildup of mucus on this apical membrane of their lung cells, okay? Now you can think about conceptually how it would be uncomfortable to have mucus kind of filling up your lungs. We also know now that extra layers of mucus might act as sort of an extra barrier for diffusion. We've thought about how that affects rates of transport, right? So this might even make it harder for gases to diffuse into the tissue. This is not good. This is not great. And the reason for this is that we have damage or deficit or mutation in a protein that was supposed to be transporting chloride, right? So in our normal tissue, right, our healthy tissue, we saw that chloride ions were being transported here to the apical membrane towards the airway lumen. But in cystic fibrosis, they're not, right? So we're not building up this solute concentration, we're not building up this higher osmolarity. And as a result, we don't have the same water movement. So in this case, as we see no chloride ions building up here, right, positives and negatives are attracted. So this also damages the movement of the sodium ions, because they should be going together. And the end result is there's nothing pulling on that water. So the mucus grows and grows, becomes thicker, doesn't get watered down. So it becomes much more difficult to clear from the lungs. And patients have to exert a fair amount of force to do this, to clear their lungs and keep them working. So finally, the last thing that we want to think about is, as we're thinking about regulating movement from external environment to the internal environment by crossing an epithelial cell layer, and therefore crossing two cell membranes, is transcytosis. Okay. So transcytosis, one of the differences here, we're going to be thinking about bigger molecules than we were in the previous example. Right, so in the previous examples, we saw ions, we saw glucose, which is relatively small. When we think transcytosis, we're thinking really big things, macromolecules. Yeah, so very, very large molecules that aren't going to fit through like a transporter embedded in a membrane. But we do have ways to move those components across cells as well. Okay, so we would do that by transcytosis. So trans, across, opposite the cell. So we went all the way across the cell. We went to the opposite side of the cell. Now, this basically involves doing endocytosis on one end and then exocytosis on the other end of the cell. So here in this image, we can see endocytosis happening at this lumen side, so the apical membrane here. And we see our exocytosis happening on the blood side, that basolateral side, the bottom of this cell, so that we get this large macromolecule all the way in. And I think in the slides, we don't have a reminder of what endocytosis and exocytosis are, but I'm going to remind you right now because I don't know when you last saw those words, but we can see the process here in these images. Right, so endo, you want to think in, right? So endocytosis is moving into the cell. Exo is out, right? So that's moving out of the cell. Those are the directions. And then the process is basically just making bubbles. Okay. So they work the same, just in opposite directions. So here we see on the endocytosis end, we've got our large particle. It comes up to the membrane. It's too big to fit through a protein in the membrane, so we don't have a carrier, we don't have a transporter. What happens instead is that we see the membrane folding in and around it, right? So you see this whole section of the membrane forming this little pocket. So this would continue, and then that little pocket would move in and peel off. Right, so now we have this little membrane baggie inside. So formally we would say that we had the membrane invaginate and then it formed a vesicle, right? But we can think about this just sort of in visual form as well. So we made a little bubble when this came up to our apical membrane. Okay. So we would then have that large macromolecule traveling through the cell in its little baggie here until it gets to the other side. And then it does the opposite process, right? So it bumps into the basolateral membrane, right? We would see it for a little while here. And then we start to see that bubble bursting, right? So here we can see where previously this would have been sealed off. So this would have been the complete vesicle. But our membranes work a little like soap bubbles. When they come into contact with each other, they merge. When this one merged, it popped, and that puts this macromolecule to the opposite side. Sorry about that. To the opposite side into the interstitial fluid. Okay. So I'm pointing out that process and just reminding you of it partially because we're going to see something kind of like this happening with neurotransmitters at the end of our next unit. So it's helpful to remember that now instead of having to remember it later on. For our purposes for this unit, we're just thinking of it as the process of transcytosis which is another type of transport. Large things involve endocytosis and then exocytosis. But in the future, we're going to talk about moving neurotransmitters, we're going to talk about vesicles, et cetera, et cetera. So that's why I'm mentioning it now, just to jog your memories. Okay. So that's it for content that you should expect to see on the exam. Okay. So a reminder of what hopefully we've been doing so far. So you've been taking a glance at slides and the textbook before class. You've been doing a little bit of pre-reading, making sure that you have some of the terms that you're going to expect to see in lecture, are familiar with them. So what I say to you isn't just a pile of gibberish. You come in, I'm explaining these things, pointing them out in images. We're doing some little practice questions. You found some way to ask content questions about this stuff. If you haven't been asking questions, a great time for questions will also be our review day on Monday. You've been doing some quizzes to make sure that you're keeping a pace with the material. Okay. And we are in our study period, so you're studying, and my recommendation to you is that you make sure to focus most on active learning techniques. So you're not just rereading and rewriting stuff that you read and stuff that you wrote. You're working on quizzing each other, quizzing yourself, doing brain dumps on whiteboards, redrawing figures, practicing problems from the book, etc. Our exam, which we'll have the end of next week. Format pretty simple. It's 100 points total. 33 multiple choice questions, an extra credit question on there. So you want to make sure before the exam that your lockdown browser is working and your computer is charged. If you have a problem with either of those things, let me know, and I will make sure we have a laptop cart here or an alternate exam format as a backup. When you get to class, you're going to put all your stuff away except for the laptop and a pen and pencil, because I'll be giving you scratch paper if you want to use it. Okay. Once we're all settled, everybody has paper. We'll do a password protected exam in that lockdown browser. When you finish the exam, you don't have to sit here till everybody finishes, you can leave. But I want to impress upon you how loud it gets in here when there's anybody talking in the hallway. It's really awkward when we can hear the echo, and it travels way farther than you think. So leave, but if you want to chit chat, go outside if it's sunny, go to another room or downstairs or something if you're going to be chatting, so that you don't disrupt people still in the exam. Final thing is I put a practice exam up, so I just want to give you some tips about how to use that. Some people find them very helpful, some people don't. So try it out and you'll see. But the way I set it up, so I'm trying to give you an idea of the distribution of questions, the kind of topics to remember. So I pulled out a question that's on there, right? So here we have one about sensors with some blanks to fill in. So what I would expect you to get from a question like this one is that hopefully you recognize this is a question about homeostasis, right? And particularly about homeostasis pathways. So you're saying, oh, okay, I got to remember homeostasis for the exam. That's something that you get from this. Hopefully you pick the right answer, which happens to be this one about regulated variables and effector organs. But you're going to go through all the answers and you're going to try them all. You're going to make sure that you understand why the other answer choices are wrong. And you're going to make sure that you understand all the terms that show up, right? So you're going to keep reading, even if you get this regulated variable effector organs one, you're going to make sure, you know, okay, I also do know what the brain is. I do also know what a set point is. I don't think receivers are real, so that one's not one. You wouldn't find it in your notes, wouldn't find it in your textbook, but you know what an error signal is. You've seen a mention of the central nervous system. So then you're prepared in case those other terms appear on the exam. That's kind of how I would go through it in kind of more detail than just taking the questions, because that basically gives you kind of more studying. They're not going to be the same exact questions on the exam. So if you just memorize the right answers, then that's going to trip you up on exam day. So don't use it that way. Use it as kind of a barometer of did I thoroughly study all the topics, and then also as a, okay, did I remember what all these words mean? Can I explain why these other answers wouldn't be any good? Okay, and that's it for today. So enjoy the extra couple minutes of sunshine. Enjoy your